BD Biosciences, a world leader in flow cytometry, has been a reliable partner to the scientific community for the past 40 years. Recent scientific advances have enabled the inclusion of mRNA expression levels in single-cell analyses. This video shows how insights can be gained into mRNA and protein expression by performing sample enrichment on FACSMelody cell sorter, followed by a multiomics analysis on the BD Rhapsody, combined in a simple, single, streamlined workflow. The BD FACSMelody is designed to make cell sorting simpler. It offers high-end sorting in an easy-to-use package for researchers. This video shows the ease of operation and convenience offered by the FACSMelody cell sorter. The startup of FACSMelody is very easy. Press the power button on the front of the cell sorter unit. It will illuminate red. Start BD FACS Chorus software by clicking the shortcut on the desktop and log in. The software has been designed with guided, simple, task-oriented screens. There are numbered tabs across the top of the workspace to indicate the order or workflow where information needs to be added. The startup process is automated. FACS Chorus software will guide you with step-by-step instructions. The upper tabs on the opening screen will serve as a guided workflow for startup. Let's get started. First, prepare a tube of CS and T beads. Click on Run Cytometer Setup. The cytometer setup is the standardized QC method to check the daily performance capability of the instrument. Load the tube of CS and T beads and follow the prompts. Click Continue. The current step with the progress bar is shown. After CS and T has completed successfully, the final step is to perform drop delay with AccuDrop beads to adjust the sort setup. Prepare the AccuDrop beads and click Continue to run drop delay. Done. Now you're ready to sort your sample. Adjust your experiment design and sample settings either from an experiment template you already used or by creating a new one. Name your experiment, give it a description, and set a sample temperature before selecting the listed fluorochromes. You can even enter the label information for each dye. Use the Acquisition Dashboard panel to load or unload samples, adjust flow rate, turn on agitation option, and adjust the gates on any plots. Click the Setup Sort tab. The Collection Setup, a pull-down menu, allows the selection of the format of the collection device, tube, plate or slides, its volume, and the sort precision mode, yield, purity or single cell. Then define the target events to be sorted into each tube and assign the sort population by clicking a tube and selecting the population of interest from the population hierarchy. Click the Sort tab and insert the collection tubes into the appropriate tube holder. It's a 5 clicks operation to get your sample sorted. Take your sorted sample and move on to the BD Rhapsody. The cartridge is a consumable and the core of the BD Rhapsody system. The BD Rhapsody Express Station is a device to hold and manipulate the cartridge throughout the experiment, while the BD Rhapsody Scanner is used to monitor the experiment through the acquisition of live images. 2 electronic pipettes are coming with the system that are pre-configured for the individual steps of the assay. Using the electronic pipette with the program optimized for cell loading, the sample is injected into the cartridge. To monitor the efficiency of this step, the cartridge is scanned just within a few minutes. The image shown reveals single cells collected in single wells. In order to minimize cell doublets, the cartridge is loaded to a maximum of 10% of its full capacity. Right after image acquisition, the cartridge is placed back into the Rhapsody Express Station and magnetic beads are loaded via the electronic pipette, now making sure that all wells are covered with one bead. After an image is taken, excess beads are washed away, and again the step is monitored by image analysis. These steps typically result in a bead loading efficiency of around 95%. To lyse the cells, a magnet is applied from below the cartridge in the Rhapsody Express Station, and the lysis buffer is added to the cells. Once cells are lysed, a magnet is applied from above the cartridge, which leads to the migration of the magnetic beads out of their individual wells to capture them at the top cover of the cartridge. A collection tube is put into place to collect the beads when washed out of the cartridge. Once this step is finished, a final image is taken to monitor the efficiency of the bead retrieval step. The collection tube is taken off the express station and transferred into a magnet, to capture the beads and change the buffer. Image analysis generates a report that outlines numerous QC metrics, including the number of viable cells captured with the bead and the observed multiplet rate as highlighted here in red. These numbers are especially informative. The number of viable cells captured with the bead serves as a good estimation for the overall library complexity and the sequencing depth needed for the experiment. The multiplet rate can provide information on the expected overall data quality. Collected beads are subjected to cDNA synthesis. The BD Rhapsody system offers two options for target amplification: a whole transcriptome amplification assay for an unbiased view on the cell's transcriptome, or a panel-based assay for detection of predefined genes to maximize sensitivity while minimizing downstream sequencing cost. Both options include library preparation compatible with Illumina sequencing platforms and using standard laboratory equipment and workflows. Data analysis is made easy by the BD-provided data analysis pipeline. Generate error-corrected expression tables from the sequencing data files without having your own bioinformatics resource, and load your data into BD's SeqGeek software for visualization. This video showcases the perfect interplay of the BD FACSMelody cell sorter and BD Rhapsody single-cell analysis system to enable sample enrichment and transcriptome, as well as surface marker expression analysis on the single-cell level. This unique and straightforward workflow helps preserve sample integrity, which is key to generate high-quality data and meaningful biological insights.